a final project for Intro to Micro. Um, I'm just doing this via a screen recording via QuickTime on a Mac. I'm sure there's plenty of equivalents for Windows if you're not running a Mac. So let's come over here. Feel free to have a fun intro slide if you would like. This is Otto doing a major chomp to Ozzy, who doesn't appear to be having a very good time. As far as this intro slide, what I'm looking for is just a title. It does not have to be super creative. Who's on the project? I think these author affiliations are pretty fun. Then getting into like the meat of the project, these are supposed to be concise and quick and brief. I don't want you guys writing a whole treatise. We're not writing War and Peace here. We were just talking about bacteria. So each isolate gets its own little time in the spotlight. For example, if I were doing this project and I had an isolate, I would need to put down this little phylogeny here with the blast matches, or the top few blast matches for my culture. Escherichia, 100% identity. I don't think anyone in the class had 100% identity, but I expect many of them to be in this like high 99s range. And so if this were the case for someone, Escherichia, Salmonella, and Phytobacter all fall under Enterobacteriaceae. And so even if this match wasn't 100%, say it was like 99.6, 99.8, whatever, we could still be really, really confident that our isolate, our culture, would also fall somewhere under Enterobacteriaceae, just because, all of the closest options fall under the same group. If, say, your top few matches actually are more scattered around the phylogenetic tree, that's actually a really interesting result. It might mean that, for example, you have some convergent evolution where the 16S gene happens to be just kind of similar in different regions of the phylogenetic tree, and then it becomes more of a debate as to where your isolate might fall. So this is essentially your attempt at constructing a hypothesis as to what your isolate might be. So the next step is to take your functional tests and compare them against known results for your blast suggestions to see if, one, it makes sense, and two, if you could maybe narrow it down to see which one is the right one. Now in this one we already know it's going to be Escherichia because the sequence I used is E. coli. Escherichia is the E in E. coli. But you should basically start by scouring the internet. Where did I put that slide? I think here's your sort of best process. So if you need to find results for each of these functional tests for your blast results, Wikipedia is your friend, right? Don't be afraid to hit a little Command F on them, find on page, and then if you can't find anything in the genus Wikipedia, try going up to family or order or class or whatever. You're very likely to find results the higher you go. However, these results are less likely to be specific to your particular genus that BLAST has suggested. For example, within a genus, it's unlikely that these traits are going to vary, but within a phylum, these traits might vary significantly. So this is sort of definitely a plan B kind of option here. If Wikipedia doesn't have anything, obviously Google is your friend. Google Scholar will get you, of course, your more academically oriented results. And then, honestly, if you're looking for like a particular functional test for a particular bacteria, or your hypothesis genus is rare or understudied, and you are having a hard time finding these, do not spend a ton of time hunting down this information. Please just send me an email. I am more than happy to help you hunt it down, or to declare it a futile search and then just maybe exclude that particular test from your analysis. I would advise against excluding a test without asking me first, though. So in this case, we know that the entire family of Enterobacteriaceae should be gram-negative, and they should be bacillus. That obviously checks out. You guys should be familiar with that based on our E. coli, the work we've done on E. coli this semester. And then you should also, on the functional test, throw in a picture of the plate and give that a comparison to a picture of E. coli you find just on Google Images. Maybe E. coli petri dish. Get the job done. Yeah, look at that. Man, look at that. Damn, they good. Be pretty careful with those Google Images results, though. You don't want to accidentally grab a picture of the wrong kind of bacteria. And then other functional tests, for example, catalase response. Within the family Enterobacteriaceae, we can't be sure of which way the catalase test will go. However, the particular genus in Escherichia is catalase positive in most cases. So I think the positive result we got sort of checks out here. But if it doesn't lyse blood, it would still check out because this trait is not conserved across all strains of E. coli. Remember, the gamma hemolysis means no hemolysis. Biofilm test, antibiotic resistance test, it's all in there. So essentially for each isolate, for each culture, each unique culture in your group, you're going to need to repeat this process. You're going to need blast results, you're going to need a little phylogeny of your blast results, and you're going to need the results of your functional tests compared to expectations that you get from published literature or the internet or whatever. So after you're done presenting each isolate and its functional traits and its blast results, you should come in here and make a phylogeny of the closest match for each isolate in your group. Essentially, this will show us how each of those isolates is related, and we can then compare sequence diversity, right, phylogenetic diversity, with functional trait diversity. This should probably be the responsibility of someone in your group that did not have a working culture. And essentially, I just want you guys to compare the functional similarities within your group and the functional diversity within your group to the phylogenetic similarities and diversity in your group. At the end here, you're going to need to cite your sources. The really important thing for citing your sources is the comparisons for your functional tests. So when you're hunting down these expectations, right? Is this blast result gram positive or gram negative? Does this blast result show bubbles on the catalase test? Does this blast result form biofilms? Right? Each of those things, I'm going to guarantee you guys don't know that. I don't know that. No one knows that off the top of their heads. You're going to have to cite your sources, and you're going to have to throw them in a little slide. In presentation settings, I prefer a link with a little blurb that just tells you what you're clicking into. Or the author's name and when they published it and what journal they published it in. Since you're going to be submitting these as a video, you're going to need to send me a link to your presentation or just a sheet with your references on it so that I can check them out, because I will. Your boy loves references. So please don't just show a video of links without me being able to click them. So FAQs, if you don't have a culture, remember, you should take this on. Also, organizing your materials for your group and polishing up the presentation, dealing with the logistics of it could also be helpful. In general, it's okay if you made mistakes at some point and you don't have a culture, or got sick, whatever. Just don't leave your group hanging. If I think I already said this actually, if your sequence sucks, we resequenced most of the sucky sequences. If you can check the resequenced folder, you might be in luck. If this still didn't work out for you for whatever reason, you can just present your functional tests, and those could potentially rule out major taxonomic groups. For example, the gram stain is just tremendously powerful in terms of determining what your bacteria can and can't be. So from what we did in the lab, we're not going to be able to get it down to a genus level, right? We don't have that many functional tests, and in this case we don't have a sequence. And so all I'm asking for is that your function has some sort of tie-in to the phylogeny of your bacteria, where you say, okay, we're gram positive, so I know we can't be, I'm pretty sure proteobacteria is, by definition, gram negative, right? So you're like, okay, we gotta, even though I don't have a sequence, I know it can't be proteobacteria because my thing was gram positive, right? That'd be a cool way to tie this in, even if you don't have a sequence. Now, if you want to go super ham and you want to dig all the way down and see how close you can get, I'll support you. I'll help you out if you want some help, but I don't think that is a very likely outcome. If your functional test results don't match your expectations, the first place you should look should always be whether your cultures were contaminated, if you made any mistakes in the lab. But if you don't have any reason to suspect that your cultures were contaminated or mixed, or you didn't make any sort of mistakes in your lab, an interesting place to go would be to test if this is true for all of your blast matches or just for one of your blast matches. That could actually help you distinguish between them if the matches, especially if the matches are not particularly closely related phylogenetically. And then I think the last thing to consider here is how conserved that particular trait is. So if you're doing something like a gram stain, that's not just going to evolve willy-nilly out of nowhere or change. Taking a gram stain, you'd be like, okay, if gram stain goes wrong, you'd be like, okay, something's weird here. If antibiotic resistance is wrong, you can be like, okay, maybe this is just a particularly resistant strain. This strain could just genuinely be more resistant than the one that was measured in whatever paper you found. You don't feel obliged to explain away all discrepancies, just present the evidence that you have and what you can find with the evidence. How are you supposed to fit this in 5 minutes? Great question. Feel free to just hit up to 1.75x speed. Boom, problem solved. I don't have to watch 10 hours of videos. Boom, problem solved. Now, between 10 groups, we'll have less than an hour videos to watch still, and you guys can say a lot more words in them. It's a win-win. I love fast videos. There's a couple groups of 4, I think, that have 4 unique cultures. If that's you guys and you genuinely cannot fit your presentation into 5 minutes, even with speed boosts, hit me up. We can work something out. I think that's it.
What? I ain't get no milk. I'm drinking the silk. I'm taking the lactase. I'm popping the pill. Eggs with the oil. What? Don't fuck with the butter. What? It comes from the udders. Yeah. That's word to the dairy-free mothers. Yeah. I ain't got no leche.